think we'll go ahead and get started. So just wanted to welcome everyone to our summer CCHABS meeting. Thank you for joining us. I am Jamie Smith from Southern California Coastal Water Research Project, and I am one of the co-chairs of CCHABS. And then one of my other co-chairs, Karen, is here. Karen, if you want to introduce yourself.
Yeah, Karen Odkins here from Department of Fish and Wildlife. And yeah, Sarah, our third co-chair today, is missing. She had a little bit of a personal thing going on today, so she won't be joining us. So it'll just be the two of us today.
Yep. So with that, you can see we already have our first speaker queued up here to talk to us today about the recent release of the National HAB Research and Response Plan. So we have Holly Bowers, who is the outgoing co-chair of the National HABs Committee from San Jose State University. So thank you for joining us today, Holly. I'll hand over the reins to you if you're ready to go.
I believe I am. You can hear me okay?
Yep, we can hear you and see the slides.
Great. Okay. Thanks for the invite this morning. I really appreciate it. And I feel like I'm on a little bit of a tour now with presenting about this report that came out, which is pretty exciting because it's really a HAB voice that has come out in this report from the community that serves as what we hope is a nice resource for a variety of individuals, no matter what you study, what angle of HABs you study, where you are in the country, what career stage you're in, or what sector you're with. So the original harness, I don't know, some of you may be familiar, but the original one came out in 2005. And so that quote unquote expired in 2015. And when we revisited that document a few years ago, we realized, wow, this really needs to be updated in a good way because we've come a really long way in HABs research and what we know. And one of the big things was that there was very little, if any, mention of freshwater HABs in the original one, if you can believe that. We used to really keep the two separate, freshwater and marine. And we've come together over the years, as most of you probably know, and now our US HAB meeting is even half freshwater, half marine presentations. So this is an updated strategy to bring that all together. And obviously there's a lot of topics that overlap. So hopefully we can move together as a giant HAB community now. Let's see. Oh, this is not allowing me to advance. Oh, there we go. Sorry. Okay, and so one of the things that we first had to do, of course, was come up with the authors. And so what we did was we thought about folks that study freshwater, marine, where they are in their career stage, what their level of expertise is, whether it's in satellites or whether it's in genetics and everything in between, and also geographic. So we wanted to get voices from all over the country, as well as a variety of sectors of people. So we really worked hard on that. So we ended up with a core of about 26 authors. And then they were given the freedom to, once they started working on their respective chapter, is to reach out to any outside individuals that they thought would be good for contributing. And so once it was all said and done, I think we had about 60 people that were involved with the final writing. And so we had conducted an initial webinar and invited the HAB community, the US HAB community. We had a survey that went out. So we really wanted to find out what are the topics that people wanted to see in this report. And I should mention that Jamie was one of the authors of one of the chapters as well. So when we sat down and looked at all the topics from the original one, also for the updated, what do people want to see now? We came up with 4 thematic areas. And so the first one was observing systems and modeling. The second chapter, so to speak, was detection and ecological impacts. And so that was really about, HAB cells and toxins and how they're moving in the environment and how we can detect them. The third focus area was HAB management, so prevention, control, mitigation. And the fourth was a big human dimensions of HAB's component. And so once everybody was sort of parsed out into their respective chapters based on their expertise, they were given 3 charge questions. So what is the current understanding and what are the advances within those topics? What are the knowledge gaps and the opportunities that we still have to look at these various avenues? And then finally, what are the future recommendations and paths forward? So those were the 3 sort of common questions that spanned all of these different chapters to hopefully bring them all together in the end as a giant document. Let's see. And so, as I mentioned before, we really want this to be a resource that a variety of stakeholders can go to and sort of get up to speed on various aspects of HABs. Also, pinpoint some collaborations that could potentially happen with those already working in the field. We really try to rely on US-based publications, or if they were from overseas, usually US folks were involved as authors, or in some cases we did include where there was some major advances that affects HABs worldwide. We did include those as well. But we're hoping that this is a document that can really be accessed and useful for policymakers, industry, research community, the health sector, regulators, managers, and even the public, because people have become way more educated about HABs, especially when they're happening right in their backyard. So really hoping this is sort of a stepping off point for some folks. And so when we got done with the writing of the whole report, we went back and sort of pulled out these common priorities, which really you could look at in a lot of different environmental fields. And so we have this table in the background material because we wanted to point out just some glaring common things. So, and most of you will probably be familiar with some of these that you've had challenges with as well. So infrastructure for various forms of data and reference materials, integration and standardization of methods across freshwater and marine water bodies, further determination of regulatory concentration thresholds for spells and toxins, support for long-term, spatially diverse data sets, efforts to address multiple co-occurring HABs, evaluation of HAB issues in the context of other stressors, increasing partnerships and coordination, and addressing the interactive effects of natural human-derived and climate drivers of HABs, especially in the context of management. So again, these probably all sound super familiar to this audience. We do have, as I mentioned, freshwater was not a part of the original harness, and so it's front and center in this new version. And so we do have this map in the introductory material, which is a summary of freshwater HABs and advisories that were reported in 2021. We also have an inset here about just showing the number of reports increasing throughout the years. We also have and I won't go into all the details of these tables that I show, but I just wanted to show that this is a resource that perhaps you're interested in, or your group, or somebody that you're collaborating with. So this is another table in the introduction for common toxin classes produced by cyanobacteria. So those are across the top there. And then we have some information about the variants within those toxin classes, some of the organisms that produce those toxins, the organs that can be impacted, and then the symptoms are listed there from exposure to those various toxins. I'm pulling out the freshwater stuff, obviously, for this group, but we have a matching table as well for marine toxins. And so I mentioned chapter 1 was observing systems, modeling, and forecasting. And so I went through each chapter and just kind of pulled out some highlights within each of them under the topics of advancements, knowledge gaps, and paths forward. But this is by no way an exhaustive list at all, because there's so much information there. I would invite you to sort of peek at the report and see what triggers your interest in sort of diving in a little more. But just to pull out some highlights to give you a taste of what's in each of these chapters. Some of the advancements in observing systems, of course, portable detection platforms have come online, satellite-based sensors, again, a long way from 2005. And we have increased our community science contributions for monitoring, which has been great. There are some knowledge gaps, of course. We have those operations and maintenance costs, those long-term sustainability issues that we always talk about. Also, there's a need for ground truthing the remote sensing algorithms to species and toxins on the ground. Some paths forward that came out of that chapter included expanding, developing, and validating new and improved technology, of course, for cell and toxin detection, because that underpins all of this, what you're observing, what goes into the models, and then how you forecast. Also, improved coordination and use of networking technologies, and the National HAB Observing Network, which I know Jamie's involved with too, is developing. That effort is coming hopefully to fruition, and will help network a lot of this information together for observing systems. Also, talking about paths forwards, having those sustained time series measurements. I know we can all benefit from that. And also the development of food web models to really understand the fate and effects of toxins. There's been work in that field, of course, but that again underlies that how we model things. So chapter 2 is detection and ecological impacts. So some of the advancements there, we've had a huge increase in the database of assays for species, genes, and toxins. We do understand a lot more about toxins in the food web, yet there's a long way to go. So you can see a lot of these are like advancements, yet in other ways there are also gaps, and we have plenty of paths forward that we can think about. And we also know more about the freshwater to marine continuum now, which we didn't understand just a decade ago. And knowledge gaps too. Toxin synthesis pathways have been slow to come online because there's a lot of work behind that. Of course, more congeners that come out from these toxin classes. Metabolomic and proteomic experiments have lagged behind sort of the transcriptional type experiments over the past couple of decades, but they're coming online. And also thinking about acute and long-term toxin exposures to organisms that are affected, wildlife and as such. And so some paths forward in this field, also reference materials that came out of chapter 1, having those sort of reference databases and infrastructure for that. So we have culture collections, but really thinking here about toxins and DNA. Also biotic and abiotic sinks for toxins in the environment, what's happening out there, acute and chronic impacts on different life stages. So sometimes, we can have lots of studies on particular species, but within that species have the life stages been looked at, and the effects from toxins. Multivariate studies are very challenging, of course. I'm trying to bring in multiple avenues to look at the synergy of what's going on. Very difficult. And also, we don't fully understand the role of bacteria and viruses in the whole bloom cycle. So there's a big knowledge gap there. Chapter 3, as I mentioned, was management, which covers prevention, control, mitigation. Just to give you a break from all those words, I give you more words. So I'm not going to read through all these tables, but what I wanted to do is highlight these 3 tables that were included in chapter 3, because I think they're a nice resource to go to get up to speed on what is being used for nutrient reduction, for algal bloom reduction, and what technologies are coming online. So this is a table that's showing the land-based and in-water body strategies for both nutrient and algal bloom reduction. So you see that those are listed in the blue on the left. And then there's some challenges and technological gaps that are called out there on the right. And I'll invite you to look at the report for the details of that. Another table in that same chapter outlines current HAB control strategies and data gaps. And so on the left, you see some well-developed and demonstrated strategies that have been talked about. The FW calls out that these are fresh water. So I'm sure many of you are aware of these and maybe even work with these different control strategies. There's some promising ones coming online or maybe used overseas in some cases, like I know clay flocculation, but not widely adopted here in the US. And then finally listed down below is the available strategies, but maybe unproven efficacy. And so again, the technological gaps are listed there for each one of those. And the final table I'll show from that chapter is one outlining emerging HAB control technologies and information gaps. So you can see they're split out by biological, physical, and physicochemical. And so some new things that they're thought about. This is not my area of expertise, but a lot of the information gaps here overlap. So we've always talked about the fate and transport of any type of method used, and how does that affect maybe benthic organisms, right? And also the cost and the feasibility and how long the treatment lasts is a cost benefit analysis. Also, of course, human health considerations and wildlife and all of that. So again, really some tables to spur on discussion, collaboration, ideas, and to think about how we'll, address HAB challenges over the next decade or so. And then the fourth chapter, as I mentioned, is about human health dimensions. And so some of the advancements under that in terms of HABs have been, of course, guidance and regulations for toxins in drinking and recreational waters. There's a national outbreak reporting system and a newer One Health harmful algal bloom system. And some of the knowledge gaps in this arena are, again, this pops up, acute and chronic exposure effects. So whether it's wildlife or humans or invertebrates or whatever, there's always these exposure effects that we don't understand the whole life cycle of. Also, there can be differences in vulnerability to toxin exposures, not just, children and the elderly, but what other maybe underlying genetics of someone makes them more susceptible or vulnerable to, say, like aerosolized microcystin or something like that. And also information dissemination has improved over the years, but there's still definitely room for improvement there. In terms of paths forward, epidemiological studies would be, of course, beneficial. Having a repository for human and animal specimens, which again, there's that theme again, right? Where we talk about like reference materials for DNA and toxins for those of us working like in the trenches of developing assays, but then also, repositories thinking about having those historical specimens that you can go back and revisit and retest, especially as new toxin congeners come online. Cross-disciplinary networks are important. I remember a couple decades ago, it was really difficult to find veterinarians that knew about microcystis blooms, right? And now I think that knowledge has gotten out there a lot better, and a lot more of them know about what it means when a dog has been swimming and drank this green water and now it's dying. And then finally, the economic, social, and cultural impacts. Sometimes we have those big events like here on the West Coast, the VA event of 2015, and that event was studied immensely after it happened, which is great. We learned from it. But also, what about those underlying bloom issues that occur over time, and how is that affecting having impacts on social and cultural aspects. So again, those are just really high level bullet points that I pulled out. I'm sure you could see some themes that kept popping up there. But I would invite you to pull the report up and get a cup of coffee and peruse through. I thought the ending slide would be perfect. The back cover of the report is a euglenoid bloom off the Santa Monica coast. And so you can find the report at Woods Hole Oceanographic Institute. So the WHOI website is where it is, because that's where our national office is. There's a research drop-down menu there, and then the harness is listed. It's the very first publication report listed there, and I just checked the link yesterday, so should be good to go. So please feel free to reach out. Again, I invite you to look at the report and see what might be of interest, and hopefully it sparks some collaborations and has us all moving in the right directions over the next decade. So thank you for your time.
Thank you so much, Holly. It looks like we have potentially a little bit of maybe a minute time for one quick question, if anybody had any questions. Otherwise, if you want to share your contact information in the chat, people could email you.
Yep, I can definitely do that.
All right, I'm not seeing any questions. But Holly is super lovely and responsive to email if anybody has questions for her later on. And then we've put the link to the document in the chat.
Oh, great. Thank you.
Yeah. So we'll go ahead and move on. It's 9:25, so we'll move on to our regional updates. And thank you again, Holly, for sharing that presentation with us. So we'll go ahead and do our kind of typical round robin regional updates. So why don't we go ahead and start with, does it make sense to start with Carly and Marissa with an overview, or should we just start from region 1? Who's ready to go?
Hey there, Jamie.
Hey.
Yeah, this is Marissa. If you'd like to start with a statewide update, I can support that. And also, Carly and I will be doing some of the regional updates for some of the coordinators that are out in the field today.
Perfect. Okay, if you're ready to go, let's go ahead and start with a statewide look, and then I see Mike will be calling on you next for region 1. So,
all right, getting set up here. And folks just trying to get the technical ones going. All right. Is my screen being shared?
it is. It looks perfect.
Okay, great.
Yeah.
All right. All right, so hey folks, I'm Marissa Van Dyke at the California Water Boards. Along with me is Carly Nilsson. We are co-leads of the Freshwater and Estuarine Harmful Algal Bloom program that's implemented statewide along with our regional boards. There's 9 regional boards and we have FAB coordinators at each. So what we're going to do is just provide like a little bit of statewide update from the program, and then we'll go to the FAB coordinators updates. Several of them have some material to share, either personally or we'll share that on their behalf. So let's get going. And please feel free to ask questions, live or in the chat box. Appreciate it. All right, just to get everyone oriented again, just join the regional boards across the state. So we wanted to highlight where we have full-time dedicated staff through our program. Again, there's limited resources at the onset, so we weren't able to get full-time staff at all regional boards, but we do have staff in the North Coast region, Michael Thomas. We have staff at the Central Valley region, Dina Schultz, and the Lahontan region. Region 6 here, Sabrina Rice. In other regions, we have part-time coordinators that help with HABS event response and communication. And as their resources are available, they can help also with the field monitoring. And you can get a hold of any of us on the California HABS portal under state agency contacts. And regarding the portal, I just want to put a plug in for this again. This is the centralized website for state of California harmful algal blooms coordination, along with many other state agencies. Agencies that prepare materials, especially outreach education to the public, are published in this portal. So we have maps that show where reports are of HABs and what their relevant advisory is or not. Sometimes we get, non-HAB reports as well that go into spill response or so maybe like a nuisance algae issue. So you can find more information there. We also have satellite imagery maps as well to help with a screening level of, like where to prioritize field monitoring, where blooms might be forming in some of the largest reservoirs and lakes in California. Again, the satellite imagery only can result the larger lakes. Then we have materials on how to stay safe for, the public, and also healthy water habits. So for areas that are not monitored, what to keep an eye out on to ensure that folks are protecting themselves while they're recreating in the warmer months outdoors. And then we also have the statewide voluntary guidance that was adopted by CCHAB network in 2016, and all those materials that support its implementation, including response plan, pre-made signs, and so forth. And then standardized procedures on how to collect samples and do that monitoring. And so if you have any questions, the California HABS program helps manage the portal, but again, we present a lot of different entities materials. So let us know if you have something to present as well. And one of the reminders that we have for folks is to please report your observations of blooms, no matter if you hear from the public, like a complaint, maybe you're Parks and Rec or Public Works manager, or water manager, or you're doing monitoring yourselves at local waterways. Please share your information. Even if you don't find a HAB, but you did do that monitoring, it's important to show where there is monitoring, where it's safe, and so forth. So please fill out our online web report form. It can intake also photos and attachments, and also if you have any illness information about fish kills and so forth. And we do publish this on the web map after we QA it. All right. We have also a little reminder about our pre-holiday assessments. This is something that's been going on for over 6 years annually. We're on our last pre-holiday assessment this month. This is for preparation for Labor Day weekend, our biggest water recreation weekend around the state. It normally has the most participation as well from CCHAB members. So really looking forward to the data that you can share with us by August 29th, again at the SINAHAB reports email. And just wanted to showcase some of the sites, the number of sites that were monitored for the previous holidays. We hope to improve this number in Labor Day weekend. So please reach out to us with your data if you have any questions as well. And in preparation for any late season monitoring, sometimes if you already signed up for the holiday assessments, if you preferred to not do the May event and do an October event, that might still be coming up for you. And then to prepare for next year, if you're interested in participating in these pre-holiday assessments or partner monitoring with us more frequent, please email us or contact your regional board coordinator. This is a QR code for our monitoring wiki. It has a bunch of resources and videos to help with training. All right, and we have some brief updates from the FHAB program on some things that will be coming from us soon to CCHAB network to review, that kind of thing. So we've been working for over a year now with SCWRP, including Jamie here, one of the co-chairs, on assessment approaches for freshwater HABs and inland waters using satellite remote sensing. So we're working on developing a report with technical advisors to document standardized protocols for processing this kind of imagery, this data, and also how to make that data open as well, meaning those open data principles. Part of this report is also to document the quality, the known quality of this data and what's unknown quality, so that we can better understand, have greater confidence in the satellite imagery based off of what we've learned, and be able to determine how we can apply the use of this data in more than screening level for field-based monitoring. So we're also documenting data calculation and metrics to help address priority management questions, again, beyond field monitoring. And we're developing some key visualizations and also an application to work with the data and look at the data just like for a single water body, a region, that kind of thing, and look at the different metrics that have been standardized. So we're really excited about this product coming out perhaps at the end of this calendar year, this report. And I'll hand this off to Carly for, well, Carly will be doing the subcommittee update in a moment. So let me go to, sorry, I can pause here and allow Jamie to go to the regional boards for their updates.
Yeah, let's go ahead and do that. So we'll just go numerically through. So we'll start with Region 1. So Mike, if you're ready to provide an update, I believe you had slides.
Yes.
Yeah.
Let me share my screen. Can you see that okay?
Yep, looks great.
Okay, good morning, everybody. My name is Mike Thomas. I am the FHAB program lead for the North Coast region. I work closely with Rich Fadness, who is our surface water ambient monitoring lead. So just a quick seasonal update. Like in years past, we have routine monitoring going on in the region. In the Klamath Basin, they're doing biweekly microsystems monitoring. And this is the tribal consortium up there. This is a map on the right showing the basin. Obviously, we have a very different system this year with the dams coming down. So looking forward to seeing how the season shakes out. Big Lagoon, we have biweekly microsystems monitoring going on up there by Big Lagoon Rancheria and analyzed by US EPA. And then the Russian River, we have biweekly monitoring going on by Sonoma Water. Pre-holiday assessments are underway, like Marissa mentioned. So far, we're all in the clear with no advisories at these water bodies. Labor Day monitoring started this week, so we'll see how that turns out. But yeah, listed here are the water bodies and the entities that are sampling. Current postings and illness reports in the region. So far, pretty low numbers. And this top table here, breaking it out by benthic and planktonic postings, and the water bodies where these have occurred. We've had some illness reports in the region. The human report on the Mad River was determined not to be cyanobacteria related. And then we have some additional human and dog illnesses that we're currently investigating. Just a reminder about the benthic monitoring recommendations that the North Coast region has provided in some of their reports. For looking at benthic cyanobacteria in rivers, we recommend the tiered approach of deploying spats as sentinel samplers. Once you see increases in those spats, conducting your visual assessments to determine cyanobacteria percent cover. And then if you do see a bloom in the river, collecting a benthic mat sample to confirm toxicity. We have brought these recommendations to the CCHAB benthic subcommittee, and we are working with them. We are working together to potentially revise the benthic guidelines, which Carly will touch on here soon. As part of that effort, Region 1 has drafted a tech memo about 2 projects we did in 2022 and 2023, evaluating this tiered approach. And then we also have drafted a memo suggesting maybe percent cover and spat thresholds. Some other stuff going on in the region. We're participating in the US EPA ROAR project for a 2nd year. Just a refresher, this project is nationwide to develop standardized benthic sampling protocols. And our project this year will focus on deploying spats in the river and seeing how many spats may be needed to characterize that river. We're also working with University of Nevada, Reno on their project looking at spatial variation and antitoxin production in river networks in California, and how various river and watershed factors may affect that. And lastly, we just wrapped up a series of partner trainings. Mid-week last week, we had our virtual training, followed by 3 options in the field at Scott River, South Fork Eel, and the Russian River. And we had a great turnout. We were stoked to have everybody in the field, and just stay tuned. I'll be distributing those materials and recordings as a follow-up once we get everything together. And as always, we very much appreciate the help of our North Coast partners shown here.
Great. Thank you so much, Mike, for that update. We'll move along. I think either Carly or Marissa are going to provide the update for Region 2, if I'm not mistaken.
Yeah, this is Marissa. I'm gonna take care of that.
Okay.
And share my screen again. All right. So yeah, I'm doing an update on behalf of the San Francisco Bay Regional Board's HAB coordinators. It's Christina Yoshida and Rebecca Nordholt. So let's get into that. So they have an update on their HAB instance so far this year. And the most recent advisories or information on reports and their outcomes are published to the HAB reports web map that's displayed right here. It's highlighting the San Francisco Regional Board area, spanning down to Gilroy, up in the North Bay. And so this reports map, just as a reminder, it shows most recent information for like the last 30 days, about four weeks, and then it fades out, and you have to go to our download section to review past information. All right, so for their updates on the incidents that come out of reports of their monitoring, so there were several reports of some suspicious discoloration in San Francisco Bay over the last 2.5 months. It has appeared to be short-lived. There was coordination for some monitoring, particularly microscopy, to understand, is this a red tide, that kind of thing, going on in preparation if in case it, it expanded in spatial extent. So there hasn't been any, major bloom of red tide, though in some patches, particularly like in the North Bay, some discrete areas have some minor blooms, and these have probably not resulted in any fish kills, unlike previous years. So far this year, San Francisco Bay has had lower amounts of blooms. Really appreciate that. So Lake Merced in San Francisco County, it's one of the highly recreated lakes with fishing, some swimming in the city that has had recurring HABs. And there is an agency monitoring that. They've been sharing their data But they've been coordinating the postings, but the data has been kind of shared with us a little late. So we're presenting what we can on our web map to alert the public on those advisories. They have been quite elevated, impacting, putting recommendations of not consuming the fish that's caught there and to stay off of boats. There has also been a few fish deaths, small fish kills in some lakes and ponds, but doesn't appear to be cyanobacteria HAB related. There's still investigation on what the cause is for those. And then for Lake Hennessy in the region, it does have an active HAB. And they want to highlight also the weekly monitoring for HABs and the data sharing from the East Bay Regional Parks District that is readily shared with the water boards, and we are able to present that on the web map. We'll pause there on East Bay updates so they can do their own update. Further, the San Francisco Bay region has been participating in the pre-holiday assessments. The sampling for those assessments has not resulted in an advisory. So they want to highlight a few of the partners. So there's some monitoring happening in Crissy Field Marsh in San Francisco County. In San Mateo County, the Marina Lagoon Beach is being monitored, and in Mountain View, the city of Mountain View, also the Shoreline Lake, which is a pretty popular lake, is also being monitored. And then in other areas in Napa County, there's another partner that's been monitoring several popular swimming locations on the river. And also in Sonoma County, there's been some other swimming locations that are being monitored by the Sonoma Ecological Center. We really appreciate our partners and really want to highlight their efforts. In particular, they wanted to highlight the Lake Merritt monitoring that's being done by the Rotary Nature Center. It's monthly sampling. And we're also supporting the lab costs for water testing. They're also collecting other water quality information to understand a little bit more about the occurrences of HABs, both marine and freshwater in Lake Merritt. Lake Merritt's in the town of Oakland, and it's connected to San Francisco Bay. And there's also in this region, there is a bay-wide coordination group for addressing HABs, particularly when there's marine blooms that come into the bay and estuaries that impact those beneficial uses there. They had a kickoff meeting for the calendar year to get everyone on the same page, all the different agencies and organizations that participate in monitoring and response coordination. And they've also updated their procedures. This is something, if you're interested in, they have made available on their website as well. And one note that one of the coordinators for the region is stepping down, but we'll continue the coordination through the rest of the year. All right, and that's the end of the update for San Francisco Bay region. And back to the co-chairs, if there's any other
Awesome.
organizations that would like to present.
Yes, I think there's some folks from East Bay Parks online. If they would like to provide an update, we would love to hear from them.
Yeah, this is Hal McLean from East Bay Regional Parks. We do have a danger. We've had it since May 23rd at Contra Loma, which is in Antioch, and our Del Val reservoir in Livermore is currently it has increased cyanobacteria, but we only have a caution due to benthic cyanobacteria. And Shadow Cliffs is at a caution, and it's been open most of the year, which is good. Quarry Lakes went to a caution from a danger in May and has been is on the verge of removing all advisories. Lake Chabot is at a danger, and Big Break, which I think is in a different region, went to a danger in July. Anya Bry has an update on Temescal and Anza.
Hi, hello, my name is Anya Bry. I'm the limnologist for the East Bay Regional Park District, and I'm going to give the updates for Temescal and Anza. So Temescal had a very severe toxic bloom for most of the year. It started in April and then finally stopped at the end of July. So we were able to open for swimming on August 3rd. We still have cyanobacteria in the lake, but currently we only see Dolichospermum and Limnorhapis, and there's no toxicity at this point, but we're watching closely. The lake is at a caution sign due to some benthic cyanobacteria in the beach area. And we have been trying some monthly dye treatments this year. Have to evaluate the outcome at this point, but a lot of people have commented to me how nice the lake looks. The water is very clear, and there's very little filamentous algae, so it looks really nice actually. And we're thinking about putting a oxygenation saturation system into the lake by the end of the year to help with the internal loading, and then to help with harmful algal blooms. Lake Anza was good for the beginning of the year, and then it closed for swimming in the middle of June. And we have a persistent microcystis bloom at this point with high toxicity. Because the weather was so warm this year, we really had difficulties keeping the dissolved oxygen up in the hypolimnium, even with the hypolinetic oxygenation system. And so we're seeing some internal loading here too, which of course is bad for any, for the blooms to persist. So that's pretty much the update, but it's currently closed for swimming at a danger level. Thank you.
Yeah, thank you for that information. I don't believe we have anyone else in Region 2 on the line. Pause. So if not, I think Marissa will be back on giving an update for Region 3.
That's right. Thanks. So I'll be doing the update for the Central Coast Regional Board of the Water Boards. This information has been provided by Melissa Daughtry. She's also a swamp coordinator, so that's a surface water ambient monitoring program. And when she has the resources and the time, she also supports our HAB coordination as well. So again, Region 3 is on the Central Coast. Let's go to our HAB reports map just to get an idea of what this area covers. So I'm just kind of zooming out on this map a little bit. For the Central Coast region, goes all the way down to Coastal Santa Barbara. There are some advisories shown in yellow, and again, the faded color shows that the updates were provided over 4 weeks ago. So let's go into what their updates are. Right, so at this region, there is routine monitoring from the ambient monitoring program called CCHAB, which includes swamp. Monitors numerous sites across the region for all sorts of different purposes beyond HABs as well. So 10 of those sites from CCHAB are focused on for assessing if HABs are present based off historical data. So these 10 sites are mostly water bodies are then included in the pre-holiday assessment. And so what's going on right now is, again, monitoring started for the Labor Day assessment, so they've gotten started with that monitoring. That's why Melissa's not able to join us today. But for the previous monitoring efforts, there has been advisories since May for Lake San Antonio. Also, it had a very severe fish kill as well. That's been investigated from the Interagency Illness Work Group. There's been some minor planktonic bloom observations that only triggered the general awareness postings for Lake Nacimiento since May, and also in Santa Margarita since late June, and also for Lopez Lake. Monitoring for Laguna Lake has shown that there is no HAB blooms currently, based off the June information, May and June information. So that's some improvement. And then also Oso Flaco, this is an estuary area, also with a stream. there has been some planktonic blooms observed there, and there are postings. And that's the extent of the updates for the Central Coast region. And this represents 2 of the counties also that help support the implementation of the holiday assessment. So back to Jamie.
All right. So unless there's anyone else on the line that has updates, we'll go ahead and move to Region 5. Dana Schultz was on the line. There she is. Has an update for the Central Valley region.
Morning. Yeah, I have some slides to share, so I'll go ahead and share my screen. Okay, are you seeing the right screen?
We sure are. Looks great.
Okay. Yeah, so I'll go ahead and share some of the HAB work that the Region 5 FHAB program is up to this summer. So for partner monitoring, we're continuing to partner with Restore the Delta. They've been monitoring the Delta around Stockton for a couple years now. They're going out, they have 7 sites along the Stockton waterfront and San Joaquin River. They're doing about weekly microcystin strip tests, and monthly samples are being sent to the lab for microcystin analysis with ELISA. And in the most recent sampling event, mid-July, we did have an advisory, a caution level advisory at a site that is just outside Windmill Cove. And you may be hearing more about this work from Spencer later in this meeting. We have another partner monitoring project that we're doing with Tulare County staff down sampling Lake Cahuilla and Cahuilla River. So there was a HAB incident in 2022. They were prepared to continue sampling in 2023, but it was a very high water year. So we're really kind of kicking off this partner monitoring program this summer. They're deploying SPAT, collecting water grabs and benthic mat samples if they observe mats, and they're going out every 2 weeks. And this project really got started at the very end of June, and up to this point, we haven't had any advisories come from this work. And they just sent samples in, I believe it was yesterday, so we'll get more results from this soon. And then I don't have a slide for this, but there's one more partner monitoring effort in Region 5. So State Board is continuing partnership with State Parks Gold Field District to sample the American River, upstream and downstream of Oregon Bar. And to this point, no advisories have come from that work either. Okay, so here's some of our pre-holiday results so far. So for a couple years now, we've done pre-holiday assessment at Hensley and Eastman Lakes, and Akira from R5 Fresno is doing the sampling there. So for Memorial Day, both lakes had a caution advisory. 4th of July sampling, Hensley was at caution, Eastman was at warning. And just early next week, Akira is going to go out and grab the next batch of samples. We supported a couple sites at Clear Lake, and then you can get the full update on advisory levels for Clear Lake on Big Valley Band of Pomo Indians webpage. But the couple samples that we supported, the suite of toxins for holiday assessment led to 1 danger, and the other 2 did not have advisory postings for July 4th, and they're doing the later three set of dates. So more to come for that. And then there was a couple additional sites that had holiday assessments for Memorial Day, Stonegate Reservoir, Pine Mountain Lake, and Lake Wildwood, and results did not lead to any advisories for those sites. Okay, and then an update on what advisories we have throughout Region 5. So I did a screen grab from yesterday, that's pictured to the right. So we have 18 sites with danger advisories across 7 water bodies. And a lot of those are at water bodies where we have regular monitoring programs, or groups are leading their monitoring programs. So you can see on the map, there's a good number of danger advisories at Clear Lake. 2 of the lakes that Hallamette, so Big Break and Contra Loma, have danger advisories, as well as a few creeks that are around Clear Lake. We have 5 warning advisories. Those are, again, at some of the places where we have a lot of monitoring, such as Clear Lake. And I forgot to mention danger and warning at Discovery Bay as well. And then we have 12 caution advisories throughout the region. And you can kind of maybe check out the map online that Marissa presented to see where those are at. And then we also have 2 algal mat alerts in Region 5 right now. One, you can see the yellow triangle up in Modoc County, and another one is down in Tulare County. And both of those come from CDFW ambient monitoring results that are shared with us. And then I'll just highlight some event response efforts we've had this summer at Discovery Bay. So the Water Board received multiple reports through our online Bloom report form that Marissa shared earlier from members of the public and residents of Discovery Bay. The last summary email I saw from the illness work group, there were 8 cases in progress from Discovery Bay that are being investigated. So both State Board and Region 5 are working on event response, and we've done 2 sampling events this summer, late June, late July. We'll likely go out again late August. There have been 13 samples total from both of those efforts. Microcystin was detected in all samples, with the highest level being during the first visit that was up in that Newport Lane, Kellogg Creek area. You can see the southwest tends to have highest advisories, and antitoxin was detected in a couple samples as well. Okay, and with that, I will pass it back to the next region.
Awesome, thank you, Dana. I realize I skipped over Region 4, but I don't believe we have any representatives from Region 4 on the line, and I don't believe that there was an update provided to Marissa, if I'm correct. So if that is correct, then we will move to Region 6, which I believe Marissa is going to be providing the update for that as well. Lots of folks out in the field these days.
Yeah, that's right, especially with the holiday assessment starting this week. Just sharing my screen, and I just wanted to take a moment. Will there be an update about the Clear Lake monitoring effort from Big Valley Rancheria, or will that be postponed this time around?
That's going to be postponed because Sarah wasn't available to join us today. So
okay, I just wanted to highlight their page, and we can put this in the chat so anyone interested can see more information about their really comprehensive monitoring program for HABs in Lake County, covering the entire Clear Lake, over 30 sites, including open water sites. And there's quite a severe HAB event happening lake-wide, particularly in the lower arms as well. Just wanted to highlight that and get that in the chat. All right, so going on to the Region 6 update. This is the Lahontan Regional Board. So I wanted to frame this for folks. So it's a region that spans from the Oregon border, the higher elevation area. There's some monitoring happening all the way up north in Eagle Lake by Susanville, and through Truckee, Tahoe area, and down south through Mammoth, Bishop, and for the south information that we have is for Diaz Lake in that region. So I wanted to showcase that extent. So for their update, there's been a lot of monitoring and partnership building in the Lahontan region. This update is provided by Sabrina Rice, the FHAB coordinator there. So Labor Day pre-holiday assessments underway. They're coordinating monitoring at 25 different water bodies. That's lakes, reservoirs, and streams with 15 partners. That's an increase. We all really appreciate our partnerships that we're able to build for monitoring, and really appreciate all their efforts on the ground. So that information will be published on the web map once it's available at the end of this month. Then they also have a special study. Again, these are field-based studies that we are able to fund with our limited resources. The special study is focused on benthic algal mats, or benthic cyanobacteria mats. The first focus study in this region on this, and they were focusing on Little Truckee River and Big Meadow Creek in the region. And so they have had some observations of benthic HABs in Little Truckee River during every monitoring event since the study has started, which is quite notable. They haven't seen these HABs forming in Big Meadow Creek, but they did observe additional blooms in another location nearby in Upper Truckee River that was downstream of their routine site. So they have also moved their sampling sites to better capture this benthic bloom and study it a little bit more closely, following the recommended procedures that we've expanded to, use passive monitoring tools like SPAT. We also do microscopy, so we get taxonomy and different toxins, and also qPCR. We're looking at toxin gene production as well when we do these benthic monitoring efforts. So for partner monitoring, she has an update that they have really focused on their partner monitoring program with Inyo County Environmental Health this year. And they are monitoring many different lakes and reservoirs, including Diaz Lake, very far down south in the Lahontan region, Mill Pond, and Pleasant Valley Reservoir. And so they're doing monthly sampling during the recreational season. And they've been really focusing on getting that training up and that partnership building and, arrangement for all the shipping for the samples. So it's been a great effort, and they hope to continue this on next year. Then they also have an update on with their HAB reports that they've received and the outcomes. So for their monitoring and also reports that they've received, there are warning advisories. So that's that yellow warning advisory for no swimming in the Tahoe Keys Lagoon in the south of Lake Tahoe, also at Silverwood Lake. There's numerous caution advisories. That's the lowest HAB advisory that we have for recreational activities. So that's in many different lakes and reservoirs. So refer you to the map to take a closer look at that. And as we spoke to, Little Truckee River does have some benthic blooms. So there's an algal alert posting there as well. And then for illness investigations, this is coordinated by the Interagency HAB Illness Work Group along with the FAB program. They have had quite a bit of illness cases. And there's many cases that are still under review involving fish, dogs, and also some human illness cases, which we'll have updates for likely more in a month or so. This investigation takes quite a bit of resources and also interviews to collect all the information to make determinations. So we'll have updates from the illness work group at a later CCHAB network meeting. All right, so that's the end of their update, and back to you, Jamie, unless there's any questions.
Nope, don't see any. And then just looking, I don't see anyone from Region 7 on the call. Okay, sorry if yeah, I don't see them. So let's go ahead and see if we have an update from Region 8.
Hi, good afternoon. I have a brief presentation I can share with.
Perfect, thank you, Kurt.
Can you see that? It's still
Yes, yeah, we can see it.
okay.
Perfect, looks.
So Kurt Larkin with the Santa Ana Regional Water Quality Control Board, the harmful algal bloom coordinator for the region. As you've seen this slide, a similar slide earlier. So Region 8 is located in Southern California, incorporates portions of Riverside, Orange, and San Bernardino counties. Just a quick overview, very high level overview of some of the bloom reports and incident responses that we've had since our last CCHAB meeting. Just kind of going from the most recent down. So we had a bloom report that came in for Big Bear Lake, where there was a report of fish deaths and a potential cyanobacteria bloom. And so in that particular case, did go out in the field and also speak with Big Bear Municipal Water District. In that case, it looks like it's not related to a potential harmful algal bloom, but high temperatures and DO levels and some emergency fish stocking activities that occurred that might have resulted in some of the fish deaths. Lake Evans and Fairmont Lake, small lake within City of Riverside, had an incident of potential bloom and observed bird mortality. In that particular case, did observe that there was significant mortality and that there was a bloom that was observed both benthic and floating algal mats. We actually partnered with the city, collected samples that were sent off, confirmed there was a presence of cyanobacteria, although they were at fairly low densities. We coordinate with California Department of Fish and Wildlife, where we had some of the ducks that were observed ill and had deceased. We actually had a necropsy that was performed on that. My understanding, the results came back that they suspect that it was the bird mortality was related to avian botulism. So that's an ongoing case that's being evaluated by CDFW, but it does not appear to be HAB related. Another incident with Cottonwood Lake, aka Reflection Lake, small privately owned lake in Riverside County. Again, reports of bloom and bird mortality. That incident does not appear to be HAB related. So it is an ongoing case, but at this point in time, it's not, we have not collected any samples from it, but we have been out on the site and walked and met with the owners of the property. Lake Paris, there's been some blooms that have come up over the last several months. Obviously, the Department of Water Resources Lake actively being monitored by DWR. There is no bloom advisory at this current time, or no advisory notifications on our. If you look at our web map, North Fork San Jacinto River, there was a bloom incident that came in regarding algal mat. Partnering with San Diego Zoo San Diego Zoo Wildlife Alliance, and we actually collected some samples, detected presence of cyanobacteria. In that case, again, the concentrations were somewhat low. It's still actively being monitored and partnered with San Diego Zoo Wildlife Agency. Mill Creek in San Bernardino County, report of an algal mat monitoring was performed earlier this year. As of the current conditions, we don't observe any issues of concern where we don't actually have any postings at the site. Mystic Lake in Riverside County, as well as San Jacinto Wildlife Area. There were some blooms that came in earlier in the year. Those particular incidents were being partnered with CDFW. There was some sampling that was done on that case. We do have a bloom that is posted a caution advisory notification on the HAB web map, but we don't have any recent data to update that. And then Lake Elsinore, partnering with the city of Lake Elsinore as well, where they currently do ongoing monitoring. They've had bloom levels that have varied from or I say currently it's at the warning level, but it has been a danger, but it's been downgraded to a warning advisory. Ongoing monitoring, so we are participating with the pre-holiday sampling, similar to the other regions. We're actually actively monitoring for at Big Bear Lake, Lake Elsinore, Lake Hemet, Mill Creek, and Lytle Creek. We're updating those results to the HAB web map. As mentioned earlier, Lake Elsinore does have an advisory that's at the warning level right now. Lake Hemet has advisory notification of a caution. And as I mentioned a little bit earlier, so we do have partner monitoring that's ongoing. Obviously, we do DWR actively monitoring for Lake Paris, CDFW monitoring for these locations as well. And some of the agencies I haven't mentioned so far, San Bernardino County does provide some data for some of their county parks. For example, Cucamonga, Gausti, Glen Helen, Prado, and Yucaipa. And then monitoring that goes on for Canyon Lake as well. That's it. So
thank you so much, Kirk. That was awesome. And it's great to see all of these partnerships that are being formed. Really cool that all the regions are cultivating these relationships. All right, so last but not least from the regional boards is Region 9. Carrie, hi.
Yeah, I do have a short presentation to share. All right. There we go. Is that working?
Looks good.
Okay, great. So yeah, just a brief update. So the monitoring that we've done this year were due to incident response and satellite notifications. So we had out of the ones that we received, about 4 needed some follow-up actions where we went in the field and did some sampling or reached out to the water body managers to get their information. And we're doing the pre-holiday assessments. We have 5 sites that are funded this year. And those are Cedar Creek Falls, San Luis Rey Rivermouth, Guajome Lake, Lindo Lake, and Spring Pond. And so Spring Pond is actually on Viejas tribal land. So they are one of our newer partners since we were talking about partnerships. And there we did the sampling with them for the first pre-holiday assessment and then handed it off to them to do the rest of the sampling for the rest of the holidays. And this year in particular, we received some notifications at Cedar Creek Falls that the water was starting to look a little green by some of the hikers. So Fish and Wildlife, U.S. Forest Service requested that we sample there a little bit earlier than we were supposed to for the Labor Day sampling. So we did that already, and then we'll just add on a post-Labor Day sampling as well for that one. And then we're in the middle of our special study this year, which is focusing on small public lakes and ponds that are in more disadvantaged communities that folks use for mostly recreational fishing, a lot of dogs and things like that. So since we haven't really monitored such smaller water bodies in the past, and we've gone out to all of them a few times, but we're waiting to sample until we actually see if there is a bloom. So 4 out of the 14 have been sampled so far this year, and 2 out of the 4 have had blooms of concern. And we will be headed out on August 26th and 28th to look at them again. So currently, Region 9 has a few ongoing blooms. One is Lindo Lake, which is at a caution level for microcystins and Cylindrospermopsin. This one is part of our pre-holiday assessment, so we'll be out there again shortly for the Labor Day. Libby Lake, this is a lake that we caught through our special study, and that's currently at a caution level right now for microcystins and anatoxin. Santee Lake, number 7, that was one that we caught through our special study. And so there's 7 lakes there. It's recycled water, but it's just the furthest upstream lake that had microcystins at levels of concern for that one. Oops. Sorry, I don't know what I just did. Heritage Park Pond, that's at a warning level right now for microcystin and Cylindrospermopsin. That is also a pond that we caught through our special study this year. And then the last 2 are reservoirs that we were tipped off because of the satellite notification and then reached out to the water body managers to find out what they were seeing on the ground and get their monitoring data. So Marine is at a caution and Diamond Valley Lake is at a caution right now too. And then I just wanted to finish with maybe more of a happy note for Lake Henshaw has been kind of our problem child, our reservoir that was in bloom and posted pretty much all year for multiple years. But finally, since November of 2023, we've had both microcystins and antitoxin A below the caution level. And right now it's been at non-detect for quite a while, and that's because they've been doing a lot of work on their end. In 2021 and 2022, they started doing some conceptual models to see how they could work with the issues there. And then in 2023, they did more monitoring and treatment, which were kind of like the shorter term minimal algicides and things like that for that year, to just do a baseline, just to kind of start to get it under control. And now they have a pilot study that they're really excited about that is ongoing. So from this year until next year, and they want to look at an oxygenation system. And they're pretty excited and hopeful that this will help in the long run, so they don't have to do as much smaller short-term treatments. And I think, yeah, that was all I had for Region 9.
Awesome. Thank you so much, Carrie. That's really cool to see what they're doing at Hensley too.
I know, finally.
Yeah. Okay, so I know we're a little bit over, but I believe I don't know if DWR would like to provide any updates. I see Brian on the line, and then I believe Karen from Fish and Wildlife had a brief update. So anything from DWR?
Hi, this is Brian Sakata with DWR. I don't have too much to add because a lot of our advisories were already covered in the regional updates. But for the Los Angeles region, I think that's Region 4, we do have 2 water bodies with caution advisories. That would be Castaic Lake and Pyramid Lake. And then in Region 5, the Central Valley region, we have 2 water bodies at caution currently. That would be O'Neill Forebay and San Luis Reservoir. And we're currently monitoring weekly at recreation areas throughout the State Water Project until Labor Day.
Awesome. Thank you for that update. And then Karen, did you have anything to
I
add?
thought I was going to give a bigger update next time. I think that's what we decided on.
Yep. Okay. Thank you for correcting me. Perfect. Okay. So we're basically still on schedule then, a little bit behind. So next we have some updates from our working groups. I'm going to pick on Carly and or Marissa, whoever, if you guys are ready to talk about the benthic work group, and then we'll just go in the order of the agenda. So mitigation and illness will be after that.
Yeah, I'll go ahead and do this one. It was actually primed really well by Marissa and Mike from Region 1, so thank you. So we started up a benthic subcommittee work group based on some concerns and issues with Region 1, because they've been doing a lot of monitoring up on the North Coast, and noticed that maybe our procedures could use a little tweaking or amping up based on some of the new information we have for the state. So we reconvened in May 2023, and we've been evaluating the most recent science and considering these revisions to the current benthic guidance. And we continued meeting actually through June of 2024 this year. We're taking a little bit of a break right now because it's the height of the HAB season. We have around 10 members, and it's been really insightful because it's been folks from Utah and US EPA folks from the Office of Research and Development. And so really great. And of course, Region 1, was spearheading this. So it's been a really great group in terms of getting a lot of expertise at the table. So we've been kind of looking at the overarching question of, the recreational use for human and domestic animals being restricted in a water body due to the presence of benthic HABs. And then also expanding the goals of the benthic guidance to be more inclusive of both immediate event response, follow-up monitoring, and then as well as some of those more routine monitoring programs. There was a pretty extensive literature review by the group, kind of evaluating existing protocols and then looking at some indicators that we want to maybe consider incorporating into our guidance. And talking about that, the indicators that we are considering are coverage, so percentage or amount of benthic mats on the ground, the cyanobacteria presence, as well as the toxins. And through all of this evaluations, we kind of realized that this is very challenging process. There's so many things to consider. The science is still evolving. So we're sort of evolving to sort of having a grab bag approach to how we would do this monitoring. So this is just kind of a good visual as to how all those different types of monitoring fit together. So as I mentioned, we had event response, follow-up monitoring, and routine monitoring, inclusive of also like when additional monitoring doesn't occur, because we often have that immediate monitoring. But because of resource limitations, we don't often get the follow-up monitoring throughout the rest of the HAB season. And then as well as having those guide the decisions of posting recommendations and the associated signage. So I mentioned this before, but these are the indicators that we're sort of, or that we are considering and incorporating in to our guidance document. We're looking at coverage, which is both percentage of coverage or presence absence. Cyanobacteria itself, both looking at the DNA or microscopy or just visual eye if you're trained, as well as those toxins, including, and this is a big one, is including the application of SPATs, which was a big recommendation with Region 1 based on a lot of their experience with a lot of their robust monitoring programs that they've had in the past. So I just want to quickly go over, I know that was a lot, but you guys we're going to have a great opportunity for you all to review these documents and consume them and present it in a much more robust way. We just kind of wanted to keep it on your radar that it's coming down the pipeline. So we are going to be reconvening with our larger group and discussing some final aspects of it come September. And then we're hoping to finalize all the products internally at the work group level, and then distribute it here to the CCHAB community for you all to review. And there's going to be quite a bit of components to that because we've been working for the last year on this. So we've got a lot of stuff together for you all. And so that'll include a technical report as well as standard operating procedures for the actual sampling, a corresponding field data sheet, and then as well as some supporting documentation as to how we got to some of these decisions points that we made, and then some different signage options to support the new guidance. So we'll be releasing those in December, and then we'll be all coming back in January at a proper CCHAB network meeting to go over comments and updates and to really vet this through. And then we're hoping before the beginning of the next HAB season that we can sort of adopt this in as some new procedures and get everyone trained up and ready to rock. So put this on your radar, calendar, however you want to do it, but expect some things coming down the pipeline to you all in December.
Awesome. Thank you for that update. Let's go ahead for the sake of time and we'll get through all the updates and then we'll see if there's any questions at the end. But up next would be the mitigation group. I believe I saw Hugh on the line earlier.
Yes, hi, good morning.
Hi, good morning.
Hello, everyone. Hugh Dalton from the Mitigation Subcommittee, giving a brief update for today. The end of last year, I reported that we were going to do some lake evaluations and do some site visits on a few of the frequent interested parties we have in touch with the mitigation group. And I'll start off with Pinto Lake in Watsonville. And I've been basically doing a bi-monthly sampling with the staff there at the city of Watsonville about every 2 weeks, beginning in January, at the end of January. Seeing 2 big blooms this year. One began in January through May, no toxins. The cyanos were Famozamenon and Microcystis, and then right after that kind of died off and went into the second bloom of the year in June. Primarily Anabina and Microcystis. Currently, the lake is closed and it has about 20 ppb of microcystin toxins present. So still doing that for the remainder of this year. A couple other lakes of note. I was able to do a site visit out at Green Valley Lake, San Bernardino County in May. and they're having a productive year of cyanos and pond weed growth. 10-acre lake there, just a few miles down the road from Big Bear. And then most recently was able to get out there to Oakland area with Bill Taylor from the mitigation group. And we did a site visit on August 1st with East Bay Parks and Stephen Burkholder, and visited Lake Temescal and Lake Anza. And I know Hal and Anya already gave updates for them this year. It's been an active cyano year, like many lakes in California. I am planning to do a larger presentation the next time we meet and give more detail and kind of do a year-end wrap-up for what we started this year, and definitely planning to continue this work into the new year as well. And I guess lastly, I want to mention Pinto Lake. One of the researchers that I met this year was Dr. John Boyle from the University of Liverpool, who's been doing some sediment nutrient research beginning in 1997. So in 2025, he plans to give a report on his findings and maybe have a little get together. Tentative name of it would be Pinto Fest, as a scientific kind of public outreach date. We don't have the exact agenda set up yet, but it's planned to be, I guess, his last visit since he's been studying Pinto for the last couple of decades, and I'll have more information on that in the new year. So that's it for me today.
And thank you for that update, Hugh. And then our last work group is the illness work group. So that would be Shannon. Let's see if I know you're having some technical difficulties. Let's see. Shannon, are you able to unmute? I gave you permissions. Maybe not.
I'm on the illness work group.
Okay.
I can try to wing something here.
Sure.
Yeah. So the illness work group, and Carly, Marissa, if you want to jump in, please feel free to winging it here. But we've seen a lot of illnesses come through this year. I think some places started reporting that perhaps weren't before. I can speak for the fish and wildlife side. We've had Shannon just rejoined. Let's see if she can talk.
She's trying again. Yeah.
Yeah.
It's like,
Okay.
I don't know why she can't.
We've seen a lot more fish kills than we did last year, and also more bird cases coming in, of course, some of which were HAB related and some of which were not. And yeah, I was going to give a little bit more of a presentation on that next meeting as well. Carly, Marissa, do you want to talk a little bit more about other stuff? Or I don't know, Shannon, if you had anything that you can.
She's alive.
I'm alive.
Oh, okay.
Hi,
Go ahead,
team.
Shannon.
I had to
good, thank you.
leave the meeting and then tag back in. When all else fails, turn it off and turn it back on, right?
Exactly. There you go.
Okay. Thanks, everyone. Good morning. Just wanted to really acknowledge all the collective efforts of the illness work group agencies, so State Water Board and Fish and Wildlife, of course, led by our lovely co-chair Karen, and then OWIHA and CDPH. Really wanted to recognize all the great efforts of our regional coordinators and their teams, because we couldn't do this without them. So it's been a very heavy season, particularly through July and August. We've had more than 40 case reports with potential illnesses. Those span everything from the humans to the dogs to the fish and wildlife cases, with a real uptick in fish and wildlife of late. So Karen's been quite busy on that front. These span across 8 out of the 9 regions. So everyone's been all hands on deck this year. Just a couple overarching notes. So again, right now the work group has been focusing primarily on acquiring the required evidence needed to interrogate these different cases. So we're focusing on the investigation part, and our determinations are coming out slowly. We have increased our meetings with the work group to weekly. And then of course is in addition to the flurry of emails that we're sending to each other and our investigation support partners, mainly the regions, on a daily basis. So please be patient if any of you are interested in what our final determinations are on all of these cases, whether they're HAB related or not HAB related. Those will be forthcoming. Hopefully as we approach fall, the case volume will slow down. We're always happy to answer questions about our process and the types of data that we consider. These are, as we perform our investigations, these are really led by our CDC guidance criteria for what they term as a HAB-associated event, whether that be the environmental data, the human case data, or the animal related data. We also consider additional things like the historical data and site assessments, especially with that institutional knowledge that's held by our water board contingencies as well as our regional partners. And then of course the professional judgment of our subject matter experts, where we really leverage all of the specific expertise from toxicology to wildlife biology, our laboratory partners, and then of course water quality being a huge component of that to inform our public health decisions. These partnerships are very critical, and they're not only critical in acquiring our environmental sampling and getting visual observations through our tribal partners in these more remote regions, especially like in Region 1 in Lahontan, but also for outreach and education. And so as part of that, the Illness Work Group will be presenting a poster at tomorrow's Clear Lake Integrated Lake Symposium. It's the first one in 25 years. And so there'll be a poster session our poster will be a courtesy of our CDFW partners that'll outline more of how our process works, along with highlighting the impact for Clear Lake cases, which represent about 10% of all of our HAB-related cases statewide to date, and that's based on our 2018 to 2023 data. So just wanted to bring those to your attention. Again, happy to answer questions. If you folks out in your field work encounter either private citizens or other entities, water body managers, county folks that have health related questions or want to learn more about what we do, please go ahead and forward those inquiries to the Illness Work Group. Always happy to assist and provide a little bit extra support there. That's all I have. Thanks so much.
Perfect. Thank you so much, Shannon. Okay, so then the last kind of CCHAB updates are from the co-chairs. So the first update was that we held a nomination period because my term as co-chair and then Sarah Ryan's term was up. We did not receive any nominations, and so Sarah and I talked, and we're both happy to continue on with another 2-year term unless there's any major objections to that. And then we've now kind of have an alternating schedule with the co-chairs. So Karen has 1 more year, and then there'll be a call for nominations at that point for that one spot, because we're going to kind of move towards a more alternating co-term, just to have continuity of previous and potentially incoming folks. So given again that there were no nominations this round, we're gonna go ahead and continue on. And again, if there's any objections to that, please reach out and let us know, and we can kind of address that. But otherwise, the other thing that we were working on was, just as co-chairs, was to reach out to the community and get an understanding of our CCHAB attendees, what their needs are, things, CCHAB has been around for a little bit. We've kind of we do things the way that we've been doing them for a long time, and just wanted to kind of touch base with the community and see how things are working, if there's things that we should maybe be pursuing more of or less of. And so Karen's going to kind of share out on the results of that survey, and then we kind of have some time at the end to maybe discuss some of the findings and some of what we as co-chairs were kind of proposing or thinking we would pursue based on these findings. So Karen, you ready to go?
Yeah. Can you see my slides?
I sure can.
Okay. Yeah, so we're running a little short on time today, so I'm thinking I'll run through these slides, and then if we don't have enough time to talk about it at the end, maybe we could stay on at the end for folks who want to chat and do a little more discussion if we need that extra time. So thanks so much for the intro, Jamie, and I'll just jump right in. If I can move the slides around. Okay, here we go. So we were very pleased at the responses. We got 68 responses, which was just a lot of folks participating. We're going to just share some of the key findings. As those of you know who took the survey, it was not a short survey. So we're really going to pull out some of our highlights of things we learned, and then we'll go into some of the action items that that we took out of that. So some of the themes of the survey that we kind of brought in and asked questions about were about audience, who's coming, where are you from, what do you do, what are you interested in? And then we went into some of the content that we have at our meetings here and the format, so what people are interested in and what delivery of these meetings are best for folks. And then, of course, we wanted to talk about the working groups and see if people like those meetings, ideas for new working groups, and how those updates are working. So like I said, you know, just the highlights here, and we would love your input at the end. So going into some of the results. So this was a surprise to us. There's a lot of people who responded to this survey who have not been coming to CCHABs for very long at all. That was the majority of folks. And then there's also a smatter, you know, about half of people have been coming for longer. But that was really surprising because we were not aware of this new community of folks coming. And as you can see in this next chart here, I think that we may have just kind of sent this out to a broader group of people. And so people who plan to start attending maybe just heard about CCHABs through this survey itself. Were actually responding to the survey. And that was about 25% of people who are answering this question are just planning to come for the first time. And then there are another group that's large that is also just, you know, have come to only a couple. So folks, we asked folks, why is attendance declining for you if it is? Most of the people who said attendance is declining for some reason, it was because of meeting scheduling difficulties. So for that one, you know, hopefully we can send out more reminders, send earlier reminders, that kind of thing to help with that. And then a lot of people also said, what are you talking about? My attendance isn't declining. And then other people changed jobs and it's not really relevant to them anymore. So those were the main reasons there. Most people are pretty satisfied with CCHAB's content. When we asked about it, you can see 1 being, I'm not satisfied at all, 5 being most satisfied. People are kind of medium satisfied to pretty satisfied. Of course, our goal is to push people towards the super satisfied side of things, but pay attention to this kind of graph layout because that was sort of the pattern that we saw with these topics as well. How satisfied are you with the research presentations? People are pretty satisfied, but could be more. Bloom event updates, same thing. Program and policy updates, same thing. And then work group updates, kind of that same satisfaction. Moving on. So as you can see here, these are graphs of how many people are actually attending the CCHAB work group. So this is for on the left for the benthic guidance, and then on the right for the mitigation work group. You can see for both of them, there's only a small sliver of people who are attending these, and there is also some overlap. And so we tend to see a couple of folks who are really invested in these. And then if we look at how relevant these work groups are to folks, they're pretty relevant to a lot of people. So it's really just about getting people to go to those meetings. And then we can see that there's a lot of awesome ideas that were written in for new work groups, and we'll talk a little bit more about that later. As one example here, this was a proposed work group that we had in the survey for monitoring equipment and talking about that and learning about that and maybe working on, some applications there. As you can see, a lot of people are super excited about this work group with 36% saying yes, 36% saying maybe, and even less than that saying no. So that's really cool. So that says to us, maybe we should have some new work groups. So in addition to that, we were asking people if they were happy with the products that these meetings provide. And people wanted some new resources to be able to kind of view these meetings afterwards and also to look at slide decks. And as you can see, it was, there were a lot of different ideas. We're going to try to provide slide decks for you, and I'll talk a little bit about that later. So we're going to try to work on this to make these more accessible to folks after the meetings. So that's kind of the summary of results. Let's jump into audience, or what our takeaways were from all of this. So like I said, a lot of new members. One of the really surprising things that I didn't know was that there's more local folks and city government folks that are attending these meetings. And then the second biggest group was state manager folks like me. So I wasn't surprised by that one, but the smaller government folks were surprising. So that's really cool. A lot of people want more tribal engagement. And we heard that from a lot of you. We also had some more tribal engagement in this survey than we're used to seeing in our meetings as well. So that was really cool. And that just really showed that we should be reaching out to those folks and really actively try to recruit them into this group. So that's something we'll be doing. In terms of reasons that people are attending, we learned that people are really excited about the research and the monitoring techniques that we're sharing in our meetings. And they're also attending because maybe they have a HAB and they're learning how to respond to that HAB and monitor that HAB. We're really happy to hear that most people are not just trying to check a box with their job duties. They are coming because they are interested. People are not attending because they are having problems with their schedules. That's the most common reason. And then I think that we can just do some earlier date scheduling and let people know earlier. And then again, like I said, more frequent reminders, I think will help with that issue, hopefully. Okay, so now let's move on to our action items from the CCHAB meeting format. So in terms of the meeting schedule, people were really split, but on what they wanted. There was the whole gamut of suggestions for that, but the majority of people still wanted quarterly meetings, so we're going to keep that format for now. We can revisit later if it's not working. And then people wanted more meeting reminders and notifications, like I said, so we're going to send out more announcements, but we're also going to try to send those like calendar attachments that you can kind of just like drag and drop into your calendar to make it easier for folks to save that and not let it get lost in their emails. And then for the regional updates, similarly to the meeting schedule, there was a really wide variety of people's opinions on these. Some people were like, just take them out. Why do we even need those? And some people thought it was the best part of the meeting and everything in between. Something that we did think was a good kind of middle ground was to develop a kind of standard almost questionnaire or Google slide or like a Google slide or PowerPoint slide so that folks can have in the regions can fill those slides out, hopefully. And then we can have something that's like a little bit easier for them to just fill in and then more standardized on the other side. And then we can hopefully share those slides out with folks at the end so that there's kind of a product in case you missed your update or this particular meeting. And then in terms of presentation availability, people were asking if we can have slides shared out. So we'll be asking presenters in the future if they're willing to share their slide deck after the meeting. And because of ADA compliance, we can't just post those on our website, unfortunately, but we will be going through a third kind of party. So it'll be like a link to a Google folder or something like that. And that's where those slides will be. So that'll be on our website in the future. And then finally, more coordination with marine HAB efforts was something that we heard. So maybe a meeting format shift where we might have more collaboration. Maybe we'll have kind of a joint meeting with marine folks, or have more, marine HAB talks or a marine theme meeting at some point, just so that we get more cross-pollination between these groups that do really similar things, but sometimes get very siloed. Okay. So now in terms of meeting content, there was interest in more periodic, like regular trainings. It seemed like there were a lot of people who are kind of new to HABs coming for the first time. And so they are interested in having kind of just a reoccurring training program. So we were thinking that we could make more fact sheets, HAB 101 trainings that are just regularly scheduled, HAB monitoring kind of trainings, and then microscopy trainings. And we'd love to hear other ideas. Of course, this is a limited capacity group, so we'll do as much of this as we think is feasible. And then speaker topics. So many of you had great speaker topics, and we are already looking into a lot of those. And we have a running spreadsheet with some of these topics and started inviting people. So thank you so much for your input. We're going to make a place where people can just throw in speaker ideas whenever they have them. If it just like comes up, you saw a great presentation, you think it'd be perfect for CCHAB, just so it's really easy for folks to do that and for us to check before we start inviting folks to meetings. And of course, just plug, we always want to hear what folks who are involved in CCHAB are doing. And if you ever have a research topic or monitoring topic that you would like to present to us, please share it with us. Okay, and last here, so we had a lot of new working group ideas from the survey that were really, really cool. And so some of those ideas, or these were kind of like amalgamations of a couple people's ideas that we kind of formed into a couple different ones, is a monitoring equipment user group, a urban rural interface kind of group, a marine freshwater collaboration group, a data management and analysis group, and a training development group. So those are just some ideas for new work groups. It seems like people were really excited about the work groups. So we decided that we were going to try out a new process for starting new work groups in the fall. We're going to ask folks to pitch their ideas in the fall meeting for a new work group. And after that, we'll send a survey with those different pitch ideas on it. And we will see who responds that they are interested in participating in these different work groups. If your work group gets more than 5 members or 5 or more members to say they will be interested, then we will facilitate just an email with getting you guys all together. And you can have a meeting and choose a kind of group champion to be the leader and come up with some goals for your group and what you would like to accomplish. And then you have a working group. So we're trying to get some new working groups going. And then we're hoping that these folks will share back at the January meeting with what they have come up with in terms of what their goals are and who's leading that group. So start thinking about those pitches, and I can't wait to hear. Yeah, okay, and we already went over this one. So I think we're at break time, and I want to make sure that everybody has a chance to go to the bathroom and everything. So maybe we can do, if you'd like, we can like stay and talk now, or we can also talk to you after the meeting is over if you have input. So let's go on break.
Yeah.
Sorry, go ahead, Jamie, if you want to.
Yeah, no, just that we're going to take a now 8-minute break instead of a 10-minute break, but we have a set of exciting speakers starting at 11 a.m. All right, welcome back, everybody. We are in the last little bit of our meeting here. So shoot, I lost my thing. We're gonna have 2 talks back to back. The first is gonna be Spencer Fern, from Restore the Delta, and they're going to talk about the current state of cyanotoxin public health engagements and overview of the Sacramento-San Joaquin Delta response to HABs. So thank you so much for joining us today, Spencer. You should have all your screen sharing privileges and everything. So I'll hand it off to you.
Thank you. I'm gonna go ahead and share the right screen, hopefully. And just making sure, I'm pretty sure everybody can see this, but just making sure.
Yes, we sure can. You're in slide sorter and
Sweet.
perfect.
Okay.
Great.
So yeah, let's see. Give me 1 second. All right, there we go. So yeah, hi, my name is Spencer Fern, and I'm the Delta Science Program Manager with Restore the Delta. Some of you probably already know me on this call, but just wanted to give I know it's already been presented, but the current state of cyanotoxin public health engagement and kind of looking at an overview of the Sacramento-San Joaquin Delta response to HABs and pros, cons, whatever ends up kind of being our problem as a community organization working in this work. So first off, I'm sure everybody on this call already knows that harmful algal blooms are a problem throughout the Delta, especially in drought years, but seems to have been a little bit less last year, and then seems to be kind of cropping up in different spots now that we're in like triple digit weather as we are right now. But harmful algal blooms do end up forming around the San Joaquin Delta in dead end areas, and these areas that also suffer from the impacts of climate change, kind of mentioned like the drought impacts. The concerns that arise from harmful algal blooms leads to impacts on fish and wildlife as well as public health. And like I mentioned, Restore the Delta has to consider the impacts of public health because there is a concern with informing the community, getting response out there in a way that effectively keeps people safe and recreating in the water. And just kind of like that overall idea of trying to keep water access as equitable as possible. And highlighting this issue that we see throughout the Delta is usually it's the disadvantaged communities that suffer from the lack of public information on HABs. And just as a definition, disadvantaged communities are communities suffering from a combination of economic, health, and environmental burdens. And if you look at the map over to the right, this kind of highlights I hope everybody can see my cursor, but this area right here is like the northern part of the Delta that we're like focused on when we're doing our work throughout the Delta. And most of the frontline Delta communities fall under disadvantaged communities under State Bill 535. So just something to note while we're talking about equity and water quality issues. We see that the disadvantaged frontline communities are the most affected due to lack of funding and lack of monitoring for harmful algal bloom threats, which is getting worked on. And obviously, I'm hearing a lot of people throughout today that are monitoring throughout different parts of California, and it's encouraging to see. And the hope is that that continues. But then in the areas where there are a lack of monitoring, it leads to insufficient public health information on harmful algal blooms. And I'm highlighting basically that if there's no public monitoring, there would be no public health engagement, which is kind of like the crux of our concern when we're working for community groups, because obviously like working to monitor is great and collecting the data is great, and then also finding more ways to engage with the public, especially in the areas where they might be more concerned with economic or other environmental and health concerns, can be kind of a hard hurdle to get over. But like I mentioned, we're collecting HAB data in the Delta. Apologies if any of you have seen this already. This is kind of a recyclement of some previous slides, but Restore the Delta has been monitoring HABs in the Delta in partnership with the State Water Board. We measure for specifically microcystis using Abraxas test strips as an initial indicator. And speaking of which, I'm currently in the lab today because right after this presentation, I'll be back out in the field. So we'll be doing that, and we're also collecting samples for that holiday assessment period, which I'm probably gonna have to touch base with on Dana for. But we do use Ben genetics for genetic testing using the ELISA plate reading methods, and kind of just highlighting like the advisory levels using the FHAB programs advisory notices. I just wanted to highlight the current signage in the Delta, and then kind of highlight where some of these these signs, although not a problem from the state side, more of a problem from the enforcement from the local side, can be kind of an issue. So we see a few limitations of current signage for public health response, and this kind of ties into the signage would only be posted if the permission of the landowner is obtained. So if there's an area where a bloom is reported, it's really up to whoever owns the land in whether or not they're going to be posting the sign, which kind of limits the public health response if it's dependent on who owns the land. Obviously, city-owned is a little bit easier to work with and easier to contact, but it can be quite difficult if the land is privately owned. And also some things that we've seen, and unfortunately, from the amount of times I've said it, at least we've never taken a picture while we're out and testing for it. But when there were blooms in the Stockton area when we were testing, some of the locations of the signs would vary based on location. And in some places it was more visible and then out of sight in other areas. And what I mean by that is one of the only public free boat launching docks over in Stockton at the Morelli Park was when there were blooms present, they put the sign at the very far end of the boat launch ramp under shade where it was hard to see. So even if people were putting their boats out, it would be the last thing they'd see because it's so far from where boats are actually being launched. And another aspect too is like when we're working in public spaces like the park at American Legion Park, which is a public park in Stockton that has direct access to the Smith Canal water, which is also a dead end area that ends up getting harmful algal blooms as it has in the past. Finding ways to put signs in places where people can actually see it when they're going into the area kind of becomes a problem and a bit of a concern. And kind of highlighting more of this issue is if there's kind of like for the landowner permission kind of needing is there's a lack of enforcement, which keeps public health agencies kind of out of this response. If it's based on a relationship between the State Water Board, government agencies, and public properties or private properties, that's one thing. But trying to find a way to get the public health agencies into the equation is kind of a problem that we're dealing with here. And ideally, with future works and maybe even some standards, I'm getting ahead of myself, that's in a future slide, there might be some ways to encourage more enforcement. But kind of the last limitation that we see in current signage is like signage translation can be lacking for some of these communities. I know there's been a few signs when they were posted out that lacked Spanish translations, Tagalog for other aspects. And I know that's something that's being worked on, but that's just some of the limitations that we've seen in the current public health response and how the signage kind of impacts that. And circling all around to kind of this idea of where we're trying to get, at least in general for all Delta communities and frontline Delta communities, is that an informed community is very important. Because if you have a community that is informed on all of the problems, whether that's health or even environmental burdens, you've got a community that's going to be stronger and better understanding of the risks. And specifically, harmful algal blooms would make, if the people understand it more, you'll see a lot more people being able to recreate safely, and then also people be able to advocate for the changes in the water that are necessary. Ideally, helping to solve both problems of the public data gaps that are arising in underfunded areas, as well as hoping that people can kind of argue for their own water right instead of becoming kind of apathetic to what's normally a poor water quality concerns in the Delta areas that they're ideally recreating in. And there is a critical need for accessible and digestible public information. And I know that this is like one of the tenets or the main ideas for the CCHAB network. So ideally, obviously, this work continues to help make it much easier and much more accessible. But regardless, it's still a bit of a problem when people have a lot of other things to concern themselves with or worry about when trying to find a way to bridge that gap, which has been a challenge altogether. And so kind of an idea we've been working with, or even I have been for like the past few years, was that the importance of educating recreators should also kind of be targeted towards the youth. And this idea is, if you inform the public from the youth side, you've kind of going into elementary schools, you're going into colleges or high schools, you could find a way to kind of increase enthusiasm from students who are learning about the waters that are kind of right in front or in their own backyard. I'm talking about like the frontline Delta communities, and I always make the joke about how in 4th grade I learned about the missions, the California missions, and it's like it almost feels like if there was a whole year dedicated to something, it could be concerns in the own Delta. And obviously, it doesn't need to be that 4th grade project, but it could be at any range of like looking into different water quality impacts in the Delta, or at least in their areas, and kind of as a way to inform from the ground up. And then ideally these enthusiasm from the students can go into the homes talking about, this is what I just learned, this is what's really exciting. And then it's kind of planting the seeds to ideally have a better future, obviously. But the end goal is to just increase that public awareness that can lead to enforceable standards that we currently struggle with in the Delta. That's going to be kind of a recurring theme for a lot of like the work we do, is like ideally we get to this point where we have standards for both recreational and for beneficial uses and for drinking water, kind of all of the above, so that we can get to a point where concerns are met. And just kind of highlighting that a lack of public health considerations for HABs will lead to lack of safe access to waters, just to kind of, and to enjoy all beneficial uses. This will then probably lead to unhealthy relationships between the community and Delta as a resource. I'm talking about people I've met myself growing up, knowing the discourse around the Delta was that it was dirty and not to jump in. At least that's what I was always told growing up. But those unhealthy relationships are very hard to overcome if they're already developed, and you've kind of got this disinterest or apathy to current public health implications due to that continued disinvestment, meaning that if people are just so used to this waterway being impacted or inundated with awful or poor water quality, you've got a community that doesn't see the benefit of a great resource like a Delta, that be able to go and fish, to be able to go swim, to be able to enjoy the cultural practices that are around the water within the entirety of the Delta. That's where it's a domino effect, where if you don't start from the beginning, you have a community that starts to kind of disinvest itself, which is really unfortunate to see from what I see out in the field testing on regular occasions. But where can we improve on public health and response? I do believe that there is room for public health agencies and government bodies to collaborate more to address the threats to the communities or the concerns. And that there's HABs research that has been done is great. And I do think there's plenty of room for more research to be done, but finding a way to use the findings in the meantime to help the Delta is a must, because at the end of the day, if we're doing research just to fund research and not have any actionable standards or differences, it almost feels like a waste of the time and effort in going into this kind of studies. And ideally, that's trying to bridge that gap between community and science and making it publicly accessible. I mentioned throughout the previous slides that standards for HABs to make public notice enforceable are also something that are very necessary, so that signs don't end up under shaded areas outside of the sight of people who are coming to recreate, and making the notices easy to understand so that community members can easily digest the information. And this also in different translated languages or whatever it happens to be, so that it's accessible to everybody who recreates in the areas. And then also making sure that the updates and response forces are equitable throughout the Delta. And this is just more circling back to how most of the frontline Delta communities are disadvantaged communities, and making sure that response forces aren't prioritized based on economic advantage or whatever it happens to be, because that's often been what we kind of experience in the Sacramento-San Joaquin Delta as a whole. And I know that was kind of a shorter presentation, but that's about all I have for that. And I just wanted to say thanks for giving me the opportunity to present here. But there's my contact information on there, and If you send me an email, feel free to answer some more questions, but there might be some time for questions, so I'll just stop there.
Yeah, there's definitely time for questions for Spencer. If there's any in the audience, we'd be happy to, or speaking for you, but happy to take questions. We have about 10 minutes or so to do questions if there are any. There aren't any from the audience. Spencer, I'm a little less I'm in Southern California, so I'm a little less familiar with all of the work that Restore the Delta is doing. But it was really cool to hear about the partnering for the sampling that you guys are doing. Could you speak a little bit about what it entailed to kind of set that up and how that partnership is working, and if you think that's like an accessible process to other organizations, like what you're doing with the dipsticks and everything?
Yeah, no, I think it's almost feels like the right place for this question because Karen was initially the first person who worked with us through the State Water Board and helped us kind of train us in setting up for HABs testing throughout the Delta, which was a great experience in and of itself. And it's been paying dividends for the interns I work with and kind of paying it forward and trying to make sure that there's more ways for this access to public testing. But to kind of talk about like the accessibility, I do appreciate, and I think that it's great and it was helpful in the time, but I know that there isn't a Restore the Delta in every Delta county, which I think is also another kind of hurdle to kind of get over for different areas. So trying to find groups that are interested, and I know that we've done some work with Shingle Springs Band of Miwok Indians and helping them set up for similarly using the same Abraxas test strips. But helping that through the State Water Board too has been something that's been helpful. But I think the problem, at least from a community access standpoint, is finding groups that are willing to do regular testing, whether that's monthly, whether that's based on visual reports that they can see. I think that's the little bit of the hiccup there. But overall, I do want to say that I think that in the event that there are groups that can be easily identifiable, I think the current steps in partnership and being able to train out is already in a good framework right now so that it could be replicatable. And I know there's the partner monitoring strategy framework that's being done by the Delta Stewardship Council and in collaboration with a bunch of different organizations. And I do think that that might even be a good resource for the future as well, at least for all of California.
Yeah. Great, that's helpful to find. Let's see. Oh, it looks like we have something in the chat, but I don't know if this was sent directly to me. Is this meant for Spencer? Adrian Sabor had put a question. I think basically they're asking about for Spencer. Great. They're asking about how your experience with the test strips. Do you find them accurate? Do you end up having to dilute samples for over 10 ppb?
So the weird thing is we haven't had a bloom in Stockton since 2022. So we haven't had any detects, which coincides with what we're seeing in the field as well as what we're sending out. So I know I'm working with Dana from Region 5 on getting some test samples to kind of test the effectiveness of the kits. But as far as having to dilute, I haven't needed to do that because in the past there were samples that went over 10 parts per billion, but at the time frame we didn't really have the equipment when we were just starting out to dilute. But I know that if we were to do it in the future, I'm sure that's something we could do. But alas, we generally save that for when we send samples off to Ben Genetics, which can do a lot more of the in-depth parts per billions, because obviously it's up to 10 parts. So that's about it. Yeah, sorry, that wasn't as insightful though.
It's okay. You can only speak from your own experience, right? So any other questions? A second. Don't be shy. Okay. I guess if folks think of other questions, Spencer has provided contact information on this slide. So go ahead and reach out if a question pops up, when you're taking a shower tonight, or something like that. And thank you so much, Spencer, for sharing this with the larger group. It was really insightful, and it's a cool model, and I hope that to see more kind of work like this in different parts of California, even in the future. So thanks for kind of being on the cutting edge of this and sharing it with this group.
Yeah, and thanks for providing the opportunity too.
Great, awesome. Okay, so we're a touch early, but I think that's probably all right. We have another talk from Blake Schaefer and Kate Myers from US EPA Office of Research and Development. So we're really lucky to have them. They're going to talk to us about a lot of the cool work with Sentinel-3 and some of the recently released kind of forecasting models that have been that was about a month ago, right? That they've been kind of coming out, and all the work behind that. And so I'm sure you have you have an exciting talk together. So I'm going to stop talking and hand it over to you guys.
Thanks so much. All right, let's see if I can. And yes, it is going to be very exciting and fascinating. Let me see if I can get my slide deck up and share my screen. Okay. Can everyone see the title slide?
Yep. We sure can.
All right.
Looks great.
Thanks so much for inviting us. So this presentation is designed just for a quick overview of our forecasting model. It does make an assumption that folks on the call are generally familiar with the satellite remote sensing, specifically with the Cyanobacteria Assessment Network, or CYAN for short, satellite data. In order to get into the forecasting model, we just won't have the time to talk about the satellite technology, but we've talked to this group and to multiple other webinars about the background of the satellite imagery in that. We're going to focus on how we might be able to provide useful one-week forecasting for the largest 2,192 lakes in the United States. So I'm Blake Schaefer, and I'm very happy to introduce Kate Myers, who is an ORISE fellow, and she will further discuss the errors in the model in a few minutes. All right, so we're going to start with a brief overview of the motivation to develop a forecasting capability, and then we'll get into some details of the first forecasting model, along with some discussion on the performance results. As research scientists, we're always looking to the future, so we're going to highlight some of our next potential steps to increase our capabilities, and then we'll conclude with a simple summary and acknowledgements. We know the timing and location of these bloom events are important, kind of like what you just heard from the previous speaker, for water quality management. And there's no real quantitative tool that exists to forecast these events across lakes for the entire US. Forecasting cyanohabs is difficult and complex because there's so many various conditions across the US for each individual lake system. We need a way to look across all lakes, which is our focus at the EPA at the moment, that could provide some level of information. And the idea here is this is a high-level model, and the hope is while this model may provide some useful information, more detailed complexities and newer models will continue to be developed for individual systems as time goes on. So we start with the relatively new updated World Health Organization guidelines. Here we're going to use their recreation alert level 1 as our action level, but other levels are certainly possible in the model. The World Health Organization describes alert level 1 as waters greater than 12 micrograms per liter of chlorophyll with a dominance of cyanobacteria. And as it turns out, the satellite algorithm that we are using in the Cyan project, is called the CI Cyano. It is measuring the cyanobacterial dominated biomass. And we have a way to convert this measure to chlorophyll A based on a collaboration with our colleagues at NASA. This has all been peer reviewed. We've talked about this before, and unfortunately, some of the stuff we just won't have time to get into too much detail today. I do want to be clear that we are talking about biomass here and not toxins. The satellite has no ability to directly detect toxins. The model framework that was chosen for our forecasting effort was the integrated nested Laplace approximation hierarchical Bayesian spatial temporal model. That's a mouthful, so we typically just refer to it as the Bayesian spatial temporal model for short. The model was used to forecast and map weekly exceedance probabilities of alert level 1 for exposure to cyanohabs for all satellite resolved lakes across the US. We are using surface water temperature, precipitation, and lake geomorphology, the lake area and depth, as predictors, since these were data sources that were relevant to cyanobacterial growth. And importantly, they were readily available on a weekly time step and at the national scale. This is not to say nutrients aren't part of the picture. We just don't have nutrient data for every single lake for every week of the year. The satellite-derived cyanobacteria detections were used to train, test, and validate the model and considered truth in observation for this study. So the conceptual layout presented here has been greatly simplified just to reduce the complexity for this talk. The full details are available in an open access manuscript listed at the bottom of the slide. The 3 model inputs included Oregon State University's PRISM Climate Group air temperature, the weekly PRISM precipitation, and the National Hydrology Dataset lake shapefile. The response variable of cyanobacteria biomass was quantified from the satellite-derived measures of cyanobacteria that we just kind of touched on. I'll note that a random forest model was used to predict lake photic zone temperature, which was previously published by our colleagues Betty Creaky and Jeff Hollister, and this was applied to this study. And it incorporates the PRISM air temperature to provide weekly surface water temperature. So again, there's no good way to get surface water temperature in every lake every single week of the year for the forecasts. So we have to drive that with this additional random forest model from the air temperature predictions to get the water temperature. So lake surface water temperature, precipitation, mean lake depth, and surface area were retrained as predictors for the Bayesian spatial temporal model. The Bayesian spatial temporal model was trained and validated with satellite observation chlorophyll A data from 2017 through 2020 for all resolved lakes across the contiguous United States. And we randomly split the data with 70% for training and 30% for validation. We then used all the data from 2021 to independently assess how the model predicted weekly cyanohabs during this calendar year. So as a reminder, we are assuming the satellite data is truth. We did do a test on how the error in the satellite data might impact the performance of the model, but unfortunately, again, that's not something I'm going to be able to have time to dive into today. But those details are certainly available if you want more information in the paper. Our model output, if we can get there, is the weekly probability for each of the lakes to exceed the World Health Organization alert level 1. So lake-wide, what's the probability of that concentration of cyanobacteria-dominated chlorophyll A to exceed that 12 micrograms per liter? So let's take a look at how the model performed during the 2021 independent assessment. And again, for time and clarity, I'm only going to focus on that independent prediction year here. So model evaluation is critical, and we all know no forecast is perfect, and all forecast models are going to result in false positives and false negatives. A range of performance metrics were included to assess the reliability of these forecasts. The summary performance metrics for the entire 2021 year included accuracy, sensitivity, and specificity all above 88%. The model's sensitivity and specificity were measures of the ability to identify the presence or absence of a cyanohab correctly. Sensitivity measured the model's ability to identify positive observations among lakes with cyanohabs, and high specificity was desirable because it indicated a low rate of false positives, thus enabling use of this model with reasonable confidence. Currently, we know the model overpredicts positive events, and the potential management expense is going to probably be something like time and costs related to additional confirmation practices, such as complementary field sampling or additional satellite remote sensing. So here's an example of what the forecast looks like, where purple colors are low probabilities and yellows are the highest probabilities. For each month in 2021, we provided just an example forecasted week. Each point is a resolvable lake, and the color indicates the probability of exceedance of a WHO Alert Level 1 cyanohab event for the second week of every month in this calendar year. So as we would expect, the highest probabilities of Alert Level 1 occurrence was in most of the CONUS lakes from July through September. That's our HAB season. But particularly in the southern, upper Midwest, and southeastern regions. I will note that high probabilities did remain in a few systems throughout the winter, mostly in the southern part of the United States. So digging into the model performance a little bit deeper, here's a breakout of the model forecast probabilities for true positives, or TP, false positives, FP, and true negatives, TN, and false negatives, FN, results summarized over this 2021 prediction year. A cutoff of 0.1 was used, and that's represented by the dashed line. It was designated for either the positive or negative predicted WHO Alert Level 1 cutoffs. The numbers in parentheses are the counts of the events in each class. So overall, the model correctly assigns lower probabilities of positive events when they did not actually exceed the WHO Alert Level 1 threshold. So now we're digging a little bit further. Here we break out the weekly model results for the true positives, false positives, true negatives, false negatives on the left-hand side. True negatives peaked in the winter, which makes sense, and then declined to a minimum in the end of July at the beginning of and through the beginning of September. Negative events by far with a dominant observation, most of the times greater than 1,000 negative events per week across all of 2021. True positives peaked during the summer through the beginning of October. Again, that's our HAB season, while true positives were lowest in the winter. Our weekly accuracy, precision, sensitivity, and specificity are shown on the right side during this prediction period. The prediction accuracy ranged from 80 to 98%, with maximums during the winter period and minimums during late summer. Precision ranged from 26 to 46%, with values below 50% occurring sporadically, mainly during the spring. Precision is related to the prevalence of bloom events in the data, where higher prevalence is going to increase precision. In general, if the prevalence was low, it would be harder for the model to achieve higher precision because there were fewer positive samples to correctly identify. And so this is the 2021 positive prevalence rate. And you can see it's relatively low. The average is 9.1%, indicating that lakes in this study have an average of 9.1% probability of an event across time. It's possible the precision may increase as the model continues to iterate training through future forecast years, and if events increase, say, as a result due to increased temperature or nutrient loading. So just briefly looking at some independent examples of the model forecasted probability versus the satellite observed chlorophyll data for 3 lakes, representing a case of low seasonal exceedance, high seasonal exceedance, and basically a continual year-long exceedance of the Alert Level 1 threshold. In these figures, the solid black line is the observed chlorophyll from our satellite data. This is considered our truth. The dotted line is the alert level 1 threshold, and the gray dashed line is the model forecasted probability. The top figure is an example from Jordan Lake. That's right near where our research facility here is in North Carolina. It's in the southeast climate zone that exhibited low seasonal exceedance from midsummer into early fall. The middle figure is from Utah Lake in the southwest climate zone with an exceedance in early spring and again in midsummer into early fall. And note that the model misses the bloom in the early spring. And Kate's going to talk about all of this in just a second. And so this is a false negative event, this miss that we had. And then we have an example of continual year-long exceedance represented by Lake Apopka in Florida. I'll turn it over to Kate.
Just to reorient ourselves, and before we get into these big picture trends of the model, I want to define the two inaccuracies that I'm going to be talking about. So the first that we already mentioned is false positives. So that is when a bloom was forecasted, but no bloom occurred. And the concerns with this is the cost associated with additional monitoring. And of the over 100,000 total observations, there were only 9,363 false positive observations in that 2021 prediction year. On the other hand, we have false negatives. So that's going to be when no bloom was forecasted, but a bloom did actually occur. And the concerns here are with environmental, human, and animal health impacts associated with not issuing an advisory in time. And there are only 1,231 false negative observations throughout 2021. And while you'll notice that there's far fewer false negatives than false positives, we are more concerned about the consequences of these unforecasted blooms. So the remainder of this talk will just be focusing on the false negatives. So I initially looked for some geographic trends in the occurrences of false negatives, and generally you can see they're clustered around the Gulf and in the Midwest. However, there are also just areas with high lake concentrations. I also looked at the trends between elevation or lake size and false negative rates, but did not find any significant relationships in the data. And while spatially there weren't any obvious trends that jumped out right away, temporally we did start to see some separation in the data. Of the 1,231 false negatives, 74% of those are what we consider a short-term event, defined as lasting between 1 and 3 weeks long. A majority of these events occurred during week 40 of 2021, which is around the beginning of October, indicating that the model might struggle with predicting short, late-season blooms. To dig into these short-term events, we categorize them within the context of what is happening before and after each negative event. So we're going to be looking at some of these same graphs from here on. So the third most common category of false negatives, making up 8% of all short-term events, is what we're defining as an early stop. So again, this is a similar illustration, the black line representing the actual chlorophyll concentration for the year, the dashed line representing the forecasted probability of the bloom, and the dotted line is placed at that bloom threshold. In this example, the actual concentration of chlorophyll stays above the threshold for 2 weeks after the gray forecasted probability falls below the line, indicating that the model forecasted the end of the bloom too soon. And below is a heat map for when this type of 1, 2, and 3 week event occurred, primarily again in weeks 40 to 45, which is roughly October and November. Ultimately, this is an error in the model that we are least concerned with. We would like for the forecast to appropriately predict the end of a bloom, to give water quality managers an estimation of when they could update the recreation or drinking water advisories. But realistically, once a bloom has been going on for this long, field teams and daily satellite imagery are going to be the primary tools for monitoring the bloom. Next, we have early stops, late forecasts, which make up 11% of short-term false negative events. These are opposite of early stops, and these occur when the forecast misses the first 1 to 3 weeks of a bloom. These primarily occurred in the summer when blooms are generally most likely to begin occurring. And finally, 57% of all short-term false negatives are completely missed events. And these occurred throughout the year, but primarily in the fall and winter. And I also want to acknowledge that 10% of false negatives were not able to be categorized as they were budding weeks where there wasn't satellite data available. As missed events were the major source of false negatives, we wanted to see if there were any substantial differences between short-term blooms the model is capturing, or true positives, depicted in green here, versus the blooms it is missing, which is false negatives, depicted in orange. The average bloom concentration was higher in true positives for 1, 2, and 3 week events, but overall we aren't finding a significant difference between the magnitude of events that the model is getting right and wrong. However, when we look at the distribution of when missed events versus correct short-term events are occurring, there's a slight seasonal shift. While the 382 missed events are mostly clustered in late October and early November, the 667 short-term correct events are mostly clustered around week 35, which is late August. So finally, I just want to leave you with 2 takeaways of this preliminary evaluation. The first is that false negatives are predominantly short-term 1 to 3 week long events occurring outside of the main recreation window for most areas in the fall and winter. These late season bloom forecasting may improve as the model trains on more data. However, it currently remains a potential blind spot in the model. And second, 57% of short-term false negative events are missed events. Other models may be better suited to capture these short-term blooms. This may indicate the lower temporal forecasting limit of the model, as the model inputs are weekly averages. This may improve if the model is trained on daily data in the future, which is something we might be considering. I'm going to skip through these 2 slides. So what's next? We would like to briefly mention our research plans looking towards the future. Our future plans include working with the U.S. Army Corps of Engineers and our existing federal partners to focus on Sentinel-2 higher resolution chlorophyll A detection. Cyan is currently at 300 meter pixel resolution, and the new work with Sentinel-2 will be 20 meter pixel resolution. Currently, Cyan can provide measures for 2,192 of the largest lakes in the US. The future with Sentinel-2 increases to over 270,000 lakes and almost all estuaries depicted in the map on the slide. While the data from the original project was valued at $5.7 million per year, the Sentinel-2 lake data is valued at $42 million per year. So in summary, a Bayesian spatiotemporal model was applied to forecast World Health Organization Alert Level 1 exceedance. We demonstrated weekly forecasts for all satellite resolved lakes during the 2021 calendar year and are working with our Office of Water colleagues on beta testing operational forecasts. If this modeling method complemented existing efforts of field monitoring and real-time remote sensing, it could become part of a more comprehensive management toolbox for monitoring water quality. A hypothetical example might include a 7-day forecast that informs managers to watch a subset of lakes with high forecasted probabilities of an event. This could be done by using daily satellite data and then complementary field observations for toxin analysis if imagery confirms the event is occurring. Sorry, the animations got a little messed up on this slide. Finally, I want to thank everyone on the team for their efforts and acknowledge that this work was funded by the EPA, NASA, NOAA, and the USGS. And future Sentinel-2 work is funded by the U.S. Army Corps of Engineers. Thanks to all of you who have interest in our work and made it all the way to the end of our presentation. We hope it was valuable and you learned something new.
Awesome. Thank you so much for that presentation. It's cool to see some of the additional work that you're doing, Kate, to look at more of the false positives. I want to, oh, and you got some clapping hands in the in the chat as well. I want to open it up and give folks a chance to ask questions. Any questions from the group? Feel free to unmute yourself and ask directly, or if you want to put in the chat, if you're feeling shy, I can read it out for you.
I have a question.
Mm-hmm.
Thank you so much for this presentation. It was super interesting, and getting those forecasts will be really, really cool. So I guess my question is, you were saying that you're gonna potentially try to do kind of like a daily monitoring effort to kind of hone in on some of that data. What would that look like, and what scale, like spatial scale, would you have to do that on to make that work?
For the daily monitoring, are you going off of what I mentioned in the
Yeah. Just like a potential project. I know that's not like necessarily definitely happening.
So I think I misspoke. It's more of we want to feed the model daily data and still
Okay.
produce that weekly forecast.
Oh, I see.
So
Okay.
it might be able to potentially correct some of those issues we're seeing where the forecast is currently a little bit bad at predicting 1 and 2 week events. So giving it daily data, it might have better temporal resolution.
So collective, like what would the collection for that data look like? Would it just be feeding in existing data sets?
When we're talking about daily data, we're talking about from the satellite.
Got it.
So,
okay. So it's not ground truthing as much.
Oh, you're talking about for field observations.
Yeah.
So typically what we use the field observations for are the validation of the algorithm to show the performance and the error metrics. And that's a little bit of what we weren't able to get into because of just time constraints. But other papers, we've done quite a bit of extensive validation work from data from the water quality portal and the National Lakes Assessment. And a few states have also given us data as well. So we're doing this with the Sentinel-2 stuff now. We're pulling in as many chlorophyll values as we can to help with validating the algorithm works both in space and time.
Okay. Yeah, that makes sense. So it's kind of two different efforts to hone in there. Thanks.
And actually, it sounded like you were about to talk a little bit more about the daily satellite observations. Do you mind completing that thought of like so you're going to potentially pull in like the daily product to provide potentially a little bit better predictions? Is that kind of what you're alluding to?
So when we started this out, I mean, we didn't think it would work this way. So we started very large scale. We pulled in the weekly composites that are made from the Cyan project. And we figured if we could just get a weekly probability, that would be amazing as it is. And so that's what this model is focused on. So now that we know the weeklies work, but we're missing, because the time step of the model is weekly, we're missing these 1 week, 1, 2, and 3 week events a lot of times because it's within the time step of the model. And so what we're thinking is now we might be able to do the same type of model approach, but give it all the daily information. And maybe we might be able to improve capturing those 1, 2, and 3 week mixed events. The concern we have, and the reason we're not convinced that's going to just be as simple, is when you have daily imagery, there's a ton of cloud cover in there. And that's one of the reasons we went to weekly to begin with, to avoid all that cloud cover. But the model handles NA data really well. So it's possible we may be able to get something working on it. The other way we've discussed it is spatially, where could we consider doing probabilities for like each pixel grid value. And so we have a new postdoc who's going to be coming on board with us, Max Beal from University of Wisconsin. And those are some of the things he's going to be working on in the coming years.
Okay. Very cool. Yeah, that was kind of what I was wondering with the daily products. And like then all of a sudden you're dealing with a lot more cloud cover. So yeah. Cool. And then there's a question in the chat from Randy Turner. Do you have a target date for forecasting with Sentinel-2 data?
I love you, Randy. So no. In fact, so we are hopeful that we get something working with Sentinel-2. So the project it's a pilot project with Sentinel-2 that is currently focused on Florida, Oregon, Ohio, and a little bit of California. I'm not sure when we're going to have something US scale working for Sentinel-2. I suspect that will, at a minimum, be a 3 to 5 year effort, depending on resources and how things go. I do think, though, once we get something working with Sentinel-2, the transfer of this model to Sentinel-2 to answer the same type of question will be relatively straightforward.
Well, that's
so to answer Randy's question, it's probably more dependent on us getting something with Sentinel-2 to work to just give us chlorophyll, but like getting the model respun up to do something with Sentinel-2, I don't think will be much of a lift. It will be almost like a reprogramming, but answering the same type of question with just higher resolution, more lakes, basically. Fingers crossed.
Yeah, fingers crossed, right? See, any other questions from the audience? Great, I don't see anything. So if you guys don't mind putting emails in the chat, because people usually think of stuff later too, just so people can contact you. Otherwise, thank you so much for the presentation. It's really cool to see what you're doing with this work, and it's cool to see the progression that the Cyan project has had in the last I realize I don't have my camera on in the last couple years, and to go from just kind of making these products available all the way to kind of having this forecast. And I like seeing the emails now, and so I'm excited to see how this develops more. So thank you so much.
Great. Thank you thanks for the invite. Appreciate the opportunity.
Great.
All right.
Awesome. All right. So with that, we have reached the end of our agenda. We are now, even though we've been running late all day, we're a little early. I did want to just pause for a second, and we didn't really have any time to take community questions on our CCHAB survey results. So I just wanted to give a second opportunity to ask questions on what we presented, what the co-chairs presented on that. And if there's no questions, we'll just hang on for a minute in case there are. But if not, thank you all for joining us, and we will see you in the fall for our fall meeting, and we will be sending you a lot more reminders. One of the things we learned from our survey, so you'll hear from us through the various listservs with that meeting date. So thank you to our awesome speakers today and everybody and our coordinators and all the monitoring, and cool to see what everybody's doing. Have a good rest of your day.